Emily is on faculty at the Africa Health Research Institute and Harvard Medical School, as well as an infectious disease physician at Mass General Hospital. She has spent extensive time doing HIV/TB research in Africa. And so we're really excited to hear you speak today, Emily.
Okay, great. Well, thanks so much. So we're here in Durban, which is the third biggest city in South Africa on the East Coast here. And that's where we have our campus at the University of KwaZulu-Natal at the Nelson Mandela School of Medicine. And then we have a rural site, a rural campus up here between St. Lucia National Park and Hluhluwe and Mfolozi National Park. So this is in a particularly beautiful corner of a completely beautiful province. And unfortunately, KwaZulu-Natal is also really one of the real epicenters of the HIV epidemics. I'm sure everyone saw this really nice paper recently that showed in great detail the HIV prevalence across sub-Saharan Africa, which shows that the HIV epidemic is at its worst along the east coast of Southern Africa and then in Botswana. But because this maxed out at a scale of 25%, it actually didn't show how really this east coast and just here south of Swaziland has truly the highest areas of prevalence. And unfortunately, that's the case in our area. So I'll show you a little bit more. But you know, this issue of HIV epidemic in South Africa, as Greg says, has been my focus now for quite a long time. I first came to Durban in 2003, which was before the public antiretroviral rollout here, and have kind of focused, you know, global health can go in a lot of different ways, but for me, I've really focused on a single problem in a single place, but I've methodologically moved around quite a bit. So one of the first projects that I did, and that's why I was mentioning Dave Murdoch, who I saw on, Dave did this project with us, was a small needle autopsy study, but we were basically trying to understand the causes of death in people who were dying on antiretroviral therapy, either early, late, or even before antiretroviral therapy. And the overwhelming message was that over half of people were dying with tuberculosis as one of the causes, and that was particularly high in the early antiretroviral therapy group. But this, of course, was then shown by many other people in much larger studies and has been shown for a long time. But we all know that tuberculosis is the leading cause of death for people with HIV. But for me, the kind of driving question is, how is it that HIV so effectively takes out the TB, the human immune response to tuberculosis, and how can we study this and what can we do about it? And also kind of trying to understand how HIV changes TB pathogenesis. So, you know, this is a hard problem to study and to figure out how to study. It's neat, there's kind of, you can argue about animal models for either disease, but they're not brilliant, and certainly there's not a great model for both together. But you know, we have observations from population level data, from epidemiological data, but I'm going to try to show you, and this is why I say just a little bit, how we're trying to set up a program where we can actually study this question all the way down to the molecular level, but also at the population level. But when I first came to Durban, I came in 2012 and moved here and moved to this KRIT, which is the KwaZulu-Natal Research Institute for Tuberculosis. And that's where I'm sitting right now. And it's really, if you haven't been here, it's a tremendous basic science facility. HHMI donated a lot of money, and in partnership with the University of KwaZulu-Natal put together this tremendous laboratory facility. And so this gave us the opportunity to start setting up a program to study lung mucosal immunology in well-defined HIV and TB patients. And we worked with the Department of Pulmonology at Nkosi Albert Luthuli Central Hospital, which is a quaternary referral hospital here in Durban, where they do bronchoscopies. And we've started out enrolling consecutive clinical cases and starting to learn how to work with the bronchoalveolar lavage cells. And we defined the TB and HIV state of those patients, but none of them were really normal, or else they wouldn't have been having a bronchoscopy. And they often had other quite complicated things going on. So we then had started a research bronchoscopy cohort. And this started about 1.5 years ago. And this gives the opportunity to try to get more precise with our definition of human disease states, so that now we can enroll healthy controls. And we define that currently as people who have evidence of latent TB infection by quantiferon and are HIV negative. And then we find people in an arm of who have untreated HIV alone, and then they go on to antiretroviral treatment. And we can, we've had so far a few people come back for their second observation once they're on suppressive ART, or TB alone, or HIV TB. And we follow the 3 diseased groups through, although that's ongoing, as I said. And this is getting better in terms of defining people and getting to this tricky question of how do you study these 2 diseases together. But it's still, there's still a lot that happens in a person who's, if you find someone who has both HIV and TB, very little about, both of these diseases have a long asymptomatic period, and it's nearly impossible at this time to tease out, when did they acquire their HIV, when did they acquire their MTB infection at first, in what order, lots of things you would want to know. Nonetheless, we have found, and I'm just going to show one slide on this because it's not the focus of the talk, but we do find that even with these patients who are not perfectly defined, but at least are pretty well defined, quite interesting insights about compartmental differences between the lung compartment and the peripheral blood. And this is just an example of one, Bongiwe Ulu, who's a PhD student in my group, who's here looking at whole compartmental transcriptional differences between bronchoalveolar and peripheral blood samples. And she's taking this forward all the way down to the single cell level. And basically finding that of course HIV has a large effect in both compartments, but that there's very little overlap transcriptionally between the effects, which actually surprised us to some extent. It's a strong type 1 interferon effect as expected in the peripheral blood, and then actually a kind of activated dysfunctional cytolytic T cell dominated response in the bronchoalveolar lavage. But I mentioned that just to so that at the end of the talk, you'll have a sense of why we set up Vukuzazi the way we did. Because in the KRITH tower over here on the left in Durban, we really have managed to set up in the laboratories and with these relationships to these clinic-based cohorts, the ability to study HIV and TB immunology quite intensively, but we're still limited to kind of small cohorts of patients people who come in through the clinics. And 2 years ago in 2016, KRITH merged with the Africa Center for Population Health in the north of the province, which is a much older institution. It's been a demographic surveillance site for since 1997. It was originally set up to understand migration patterns and migration for work and the impact of that on rural South Africa in post-apartheid era. Then, of course, being in this epicenter of the HIV epidemic, it went on to study kind of the demographic impact of HIV and then HIV treatment. But it's really been its roots are in demography. So when we merged these two quite disparate sets of disciplines 2 years ago, we wanted to launch a program that would allow us to really start to bring these two together, and also to kind of challenge us from both sites to start to think and really put our focus on using the science to improve health. So we kind of sat back and looked and we said, okay, we know we're at the epicenter of the HIV and the TB epidemics, and there's also this rising non-communicable disease epidemic that's coming through the African continent, and we're probably at one of the leading edges of that in South Africa. So, if we try to figure out scientifically and public health-wise how to address those questions, and there are lots of them, but really the main question is, can we figure out a way to harness the kind of genomic, modern computational biology type of science and put it together with population science and actually use the insights from these to improve health in rural Africa. And how can we design a program to jumpstart that? And so that's where we sat down and we designed the Vukuzazi program. So the name Vukuzazi means wake up and know ourselves in isiZulu. And it has both a kind of individual as well as a collective sense to the word. And so what we decided to do, and we thought quite hard about, I mean, we wanted to kind of get a quick sense of all the major diseases, ideally, in the population that were causing morbidity. But we had to look carefully at the list, and we also felt we had a responsibility to any diseases that we identified to deal with clinically in a responsible manner. So we had to cut certain conditions like mental health off for the moment because we didn't have capacity to deal with them. So we decided to focus on HIV, tuberculosis, diabetes, and hypertension to start, as well as collecting information about important risk factors like nutritional status, tobacco and alcohol use, and then some about environmental exposures. And then at the same time, we wanted to collect a suite of biosamples that would support the kind of scientific approaches that we wanted to be able to take to understand the determinants, the acquired and genetic determinants of these conditions. But we put our investment initially in this definition of the precise human phenotypes and the sample collection. We didn't have funds to support all of this, these next generation science approaches, although we've started to write for grants for those, and that's a space that we hope to get into. But the idea was to establish a platform for what we call next generation population science that would allow us to identify and then start to address the local questions, which we hoped would be of global relevance, and then very quickly to move towards testing interventions to improve health. So how does it actually work? We decided to stick with the traditional demographic, within the traditional demographic methods of the site, because remember, for 15 years, they've been surveying the population in their homes, and HIV has really been the main focus and the only focus in terms of disease definition. But it does mean that, for example, for HIV, most of the people who've had incident HIV over the last 15 years, the date of that incidence is known. So it was very important to be able to link what we did with Vukuzazi to that wealth of demographic and HIV information in the database. So we recruit people in their homes and make sure we establish their link to the demographic surveillance. But then we started a new feature, which was these screening health camps that allow us to do measurements, and we have a chest x-ray van, so we do chest x-rays there. We collect samples that we transport to the laboratories, and we do some clinical labs to define the health state of the person, and then we do biobanking. All of the information from the chest x-ray and all of this flows into a data system that allows our medical officer to review every single case, and then he brings the challenging cases to our clinical review team, and we discuss them and figure out what to do. And then we manage the results of those in a more individualized fashion. So the first, on the left of the screen, it's all quite the same process for everyone, because we really wanted to have consistent information. But then, of course, once we know what's going on with each individual person, it becomes individualized. So then we go back to people's homes, and of course, if everything's normal, they get those results by SMS. But if there's anything that requires a repeat measurement, like a second blood pressure measurement to decide if someone actually has hypertension, or a blood glucose measurement to harmonize, because we check a hemoglobin A1C for diabetes, but we need to harmonize it into our Department of Health care pathways. So we do a fasting blood glucose at the home. And then after those confirmatory tests, and if anyone needs referral, then people are linked into the public health system, either at the primary healthcare level. There's 11 primary healthcare clinics within the demographic surveillance area, and we have an RE nurse in each of those, so we can absorb that without overburdening the normal DOH functions. Or if necessary, we can actually refer people to the referral hospital in Tlabisa, or even down to Durban, in some cases to the pulmonology department in Kosi Albert Lutuli Hospital. So the results of all of these end up coming together into a large integrated clinical and biological data system that I'll show you the results of in a second. So we've been on a very rapid timeline with this project, and all of this is not yet published, and I'll tell you where we are. But we started, I think we wrote the white paper about this in like May of 2017, and we got the protocol approved in November of that year, and we started operationalizing it right at the beginning of January 2018, and we launched the pilot at the end of May 2018, and we enrolled quite slowly. We did a slow pilot to the first 1,000 people, and then we had a pause where we adjusted the protocol based on the initial results because we really didn't know what we were going to find. And then we adjusted the protocol, and then post-pilot we started to ramp up enrollment, and we've been at about 100 people per day since 2019. So we had Vukuzazi's first birthday just a couple months at the end of May of this year. And for the moment, we continue to have the ongoing population platform also running parallel because we didn't want to risk kind of messing that up, basically, if we weren't able to go as fast as they were. And then at the same time, we're trying to establish longitudinal cohorts that are based in the clinics to capture incident disease, because obviously, you know, incident tuberculosis, incident strokes, cardiac events, HIV infections, you know, it will be important to better define these baseline samples. And then all of this was actually designed with this goal in mind, which was to start large-scale HIV and TB incidence interventional studies from 2021. So we wanted to establish this information as quickly as possible and then allow ourselves to plan this, and that's synced up with our welcome renewal. So just to give you some images of what this actually looks like, we're out here in rural KwaZulu-Natal. There are lots of cows. There's a video that you can find on the RA website, and when I first saw it, I said, that is too many cows. It's like cow porn or something. But then every time you go to Vukuzazi, there's always tons of cows. So it's actually pretty accurate. But what's quite cool is there we are, but we've established, one of the trucks has a solar panel, solar powered WLAN network. And we have this really high-tech data system that goes right out into the field. And as I said, you know, it's critically important to ensure that we have the right person who we know their unique identifier for the demographic surveillance data. So we go through, we establish that we have the right person, and then we use a wristband system when they're at the camp so that every single piece of information or sample that they give is scanned onto the wristband, because, you know, we obviously couldn't start the study until we had confidence that, you know, each of these tubes of blood that we're drawing out in the middle of rural KwaZulu-Natal, we would link up to the right person and we'd eventually get the right lab result to the right person. And these are the samples that we're taking and the tests that we're doing. Everyone has an HIV ELISA. People who are HIV positive have a viral load and a CD4 count. And everyone has a hemoglobin A1C. We store plasma, Buffy coats, and serum on everyone, RNA patch gene tubes on everyone, rectal swabs on everyone, and then the TB the TB sputum is collected on people who have an abnormal lung fields on their chest X-ray, or answer affirmatively to any of the 4 TB symptom screening questions. So that's the WHO TB prevalence survey strategy 1. Basically, we tried to make the TB strategy linked up with the WHO prevalence survey methodology. But any sputum that we have, we then use GeneXpert Ultra as well as a midget test on. We do both. We tried to stop one or the other, but after our pilot, we saw that that really wouldn't work, so we continued doing both. We do drug susceptibility testing on anyone that's positive, and we try to store aliquots. I mean, we definitely store aliquots of any strains that grow, obviously. We try to store aliquots of some raw sputum, although the sputum volumes are not always sufficient for that. It's critical to log the specimens, you know, to have a chain of custody of the specimens, and that starts out in the field. And then we had to set up a processing laboratory up at the Samkele site. where samples are aliquoted, and then there's a lot of driving back and forth between Sumkele and Durban for the samples and for all of us. I just did it earlier today. So I was there this morning and now back in Durban now. In my opinion, the crown jewel of Vukuzazi is this digital chest x-ray van. You're kind of in the middle of a field surrounded by a lot of trucks, and you walk into this van and you see the most spectacular chest x-ray. This is the van that has the solar panels on the top and hosts the local wireless network. And the chest x-rays are done there, and then you're in this little ante-room and you can see the chest x-rays. The radiographer is excellent. And then the images immediately goes into this image analysis, CAD for TB. We have the 5 version, and gets an automatic score. We had hoped that the score would allow us to triage a little better than it has worked out to do, but we're still working on that. But we had to lower the threshold quite a bit. So we're actually taking sputum on a fair number of people who end up having pretty normal chest x-rays, but we found if we used too high of a threshold, we were missing a lot of chest x-rays that later turned out to have lung-filled abnormalities. So that's been a little challenge, but in the end, we're collecting more sputums than strictly needed, but that's been good. And then eventually the chest x-rays are read, they immediately go to the cloud, and then we have a radiologist in Durban who reads them, and we can all see them in that clinical review tool to help with the patient's disposition as we go. So I said we had our first birthday right at the end of May, and it corresponded with enrolling our 10,000th participant. So Vukuzazi is going at quite a high rate, and that was May 21st. So the TB cultures for that first 10,000, which is going to be the first set of patients that we formally report, I think they've just closed. So unfortunately for the results I'm going to show you today, I don't yet have ready detailed TB epi, but I'll show you some, and then watch this space for more on our active TB cases. But in terms of the results, the early results from Vukuzazi, I mean, as they've been coming in, we see it is apparent that they fall into different categories. So the first one is obviously because it's a population-based cohort, we can talk about population epidemiology. We know the denominator, we know who participated and who didn't, and we can talk about infectious disease, epidemiology, non-communicable disease, risk factors, overlap of disease, spatial analysis, cascade of care. I'm only going to speak about these 3 today. Much of these analyses are still ongoing. But separately from that, and I think also really interesting, is we can kind of only worry about the people who we did enroll, but go quite in detail into these detailed pictures of human disease in its states of overlap or in its detailed states. So I'll show you 2 examples of what I mean by this. One is, we can look at detailed HIV phenotypes, and I think we can see some insights into transmission and viral evolution there. And then we can look at TB phenotypes and maybe start to see some insights into pathogenesis and immunity. So this is what the enrollment statistics look like for the first 10,000 participants in Vukuzazi. So these are the dates. We enrolled this over 1 year. And to be eligible to participate in Vukuzazi, you are a confirmed resident of the population of the demographic surveillance area, and you're 15 years or older. So to get to the 10,071 people who were enrolled on the day when we enrolled 10,000, there was a denominator of 17,189 eligible people. So we go and we try to contact everyone, but even some eligible people who we confirm to be alive, resident, and of the correct age, we still can't contact. So that's about 13% of all the people. We contacted 87%. And of those, about 1/3 versus 2/3 participate or don't. So 1/3 of those contacted don't participate. And that's about 4% of them actively refuse to participate, which is great. They hear about the whole study and they say, no, I'm not interested. And that's good because you want some people to hear about it and not want to do it. But the larger proportion, 29% of people who contacted, say to our recruiter, yes, I'll come, but then they haven't come yet. So they're actually still eligible to come. But I think probably most of those people are voting with their feet, but it's also possible that it's inconvenient for some people. And we're still working on that, of trying to increase our participation rates. But so 66 or 67% of people actually come. But the good news is of those who come, almost all participate in all aspects of the program. So 100% of people participated in all the body measurements and questionnaire, 99.5% give the blood samples, 99.7% give the rectal swab samples, although I have it from the lab that about one in 10 of those don't grossly look used. And then almost everybody does the chest x-ray except for the pregnant women who are triaged in a different direction. They just give a sputum. So we're happy with that people who do participate are being phenotyped kind of in completion. And here you can see, and I'll be quick, but you can see that we do have different rates of participation across age and sex categories, with generally higher participation by women at all ages and lower participation in working age people, especially men, higher participation in older people. But overall, 66% of the total population is participating. So with that in mind, we can just look quickly at the population epidemiology. I'm going to show you a number of slides that are all kind of the same type. So I'll explain the first one and then I'll go quickly through the rest. But these are for each disease. In this case, it's diabetes defined by hemoglobin A1C greater than 6.5. We have weighted for non-response using age and sex categories as shown. So we have the under 25-year-olds, 25 to 44-year-olds, 45 to 64-year-olds, and over 65-year-olds. And then this is disease prevalence in the population. So for diabetes, you can see that it's increasingly prevalent with age, and in this middle age group, it's significantly more prevalent in females than males. Hypertension has a similar pattern of increasing with age and being more prevalent in females than males. These numbers are based on the first blood pressure measurement at the camp only. Of course, that's 3 different measurements taken 5 minutes apart, but it's on one date only. We do, as I said, go back and do a second confirmatory measure on a different day, but that's not what I'm showing you here. So this is the WHO STEPS definition. So this is probably a little higher than what, people who truly need hypertension treatment. So our HIV prevalence is striking. Always just kind of pause on this slide and just take a minute to see that, over half the women in this middle-aged group are HIV infected. And even it ends up kind of looking low, but the men of the same age group and the women and men in the next age group, about over a third are HIV infected. And that's, those are the high rates that we're experiencing in this area. And of course, some of that is from surviving on treatment, but nonetheless, those are high rates. Then this is an interesting definition. So here we're trying to get an idea of the people who have had TB in their lifetime. And people will have seen there was just that interesting paper about the increased risk of mortality for people who have had TB in the past. So this is kind of the idea that, although TB can be thought of as a single event disease, it does have sequelae. And it's interesting to know who's been affected by active TB in their life. Here we're combining historical data, so people who tell us that they've had TB in their life, as well as the chest x-ray data, people who have evidence of TB on their chest x-ray, and I'll come into that in a little bit more detail. But this is interesting because it shows a different pattern of disease with males more than females affected, especially in the older age groups. I'll go very quickly through these. You can see we have an obesity problem, especially in females, smoking, especially in younger males, as well as drinking with the same distribution. Of course, for both of those, the frailties of self-reported data known, but so these are probably underestimations. Okay, so time is going, so I'll move quickly on to this other way of looking at the data, which is now getting into not worrying about the people we didn't see and whether we need to wait for non-response or think about how the population level data may be biased by response, but just focusing on the people that we did see, but getting into more detail with their phenotypes. So here, we're looking at HIV status, and we can define people as either having newly diagnosed HIV. So we diagnosed their HIV infection at their visit to Vukuzazi for the first time. Then there's people who are HIV negative, and that's blue. Then there's people who know that they have HIV, but they're not on antiretroviral therapy, and they're viremic, which is a small number of people. And then there's kind of 4 different flavors of people who have HIV and are on antiretroviral therapy. The yellow is people who are on suppressive effective antiretroviral therapy, and that's, as you can see, by far the largest group. And then there's people who are failing therapy at either low or high levels. So what's, I think you can see from this way of looking at the data is, first of all, this interesting thing about looking all the way across the lifespan. You can see we have like many 90-year-olds in the cohort. You can also see this, extremely high rates of HIV prevalence in these middle-aged age groups. But also it's quite striking that most people who are HIV positive are clearly well suppressed on antiretroviral therapy. And it makes you wonder, how is it that we still have such ongoing high incidence rates? Because we do. But if we look just at the HIV positive people, you can your eyes drawn to this side of the graph, where although, as you saw before, there were actually fewer, the HIV prevalence was lower among these age groups. Among HIV positive people, for example, here, men in their 20s, half of them are viremic, which is quite in contrast to like 50-year-old males. So for a kind of precision public health set of questions, this points us towards focusing on these younger men and women to focusing our prevention efforts. But what I also find interesting about looking at the data this way is, for a disease like HIV, where we've had excellent diagnostic tools like the HIV ELISA and the HIV viral load for many, many years. And so we all share kind of an excellent conception of HIV pathogenesis in humans. And we know the history of the disease in this setting. When we look at a graph like this, we can actually start to come up with ideas that we can actually see, for example, features of the virus in this data. So, say we look at these 50-year-old males, well, most of them are on well-suppressed on antiretroviral therapy. But for example, these ones who are failing therapy, we know the history of antiretroviral treatment in South Africa, and we could even hypothesize that we could imagine what mutations their virus might have in it. And actually, we've started to do just a little bit of viral sequencing, and if you thought that maybe there's K65R in there, you're right. We can see that that's true. Whereas, for example, these young men over here who are failing therapy at high level, we think, what's happening with them? Well, they're probably actually perinatally transmitted adolescents who are now growing into adulthood and failing therapy that they've been on for quite a long time. Maybe they have protease inhibitor mutations, and in fact they do. So just to make the point that with this kind of numbers and with this kind of data across the lifespan, we can start to see things about the pathogenesis of the disease in the population, even before we get into the biological and omics analysis. And we have such granularity, because the numbers are quite large, we have a lot of granularity in the data. So this is the same data, but now just for females, and each year of age is given its own bar here. So we have our 15-year-olds through our 101-year-old. And just a different way of looking at the same data, but I think what we see here is, again, seeing that we have representatives from really elderly ages in the cohort, that we have a handful of quite elderly people living with HIV now, but we see this really large peak here. And I think for me, at least when I look at this, I start to imagine what's going to happen in 10, 15 years. This generation is going to be aging with their HIV, and that raises all sorts of questions. But obviously, at least to me, the most striking thing about this graph is the slope of this line here, which is just terrifying to imagine being one of these 15-year-old girls who really, there are very few of them who are HIV positive at this time, but they're entering into sexual debut. And if their experience is going to be the same as what the generation above them has experienced, their risk of HIV by acquisition by the age of 30 is just huge. And so this kind of demands that we address that. But now I'm going to go on to talk about tuberculosis. So I just have this one slide about TB in the population, and I just want to share the idea that, you know, for now, we looked at HIV, which is, as we said, we all share a good understanding of it because we have excellent diagnostic tools. So now if we look at TB, a disease for which we don't have excellent diagnostic tools and which we don't all share a really good conception of the pathogenesis of the disease in humans for that exact reason. But what if we do the best that we can and then with the same large numbers, look across the lifespan, and does this give us any insights actually into pathogenesis? So here, just to explain these categories, the red is active TB, and this, of course, we could get more detailed, but here I've lumped together microbiologically proven and radiologically proven active tuberculosis. We have people who have no evidence of prior TB, and then we have 3 flavors of people who have evidence of previous TB. There's people who both tell us that they've had previous TB and been treated for it, and whose chest x-rays show scars of that. There's people in yellow who just tell us that they've had TB in the past. And then there's people who actually don't tell us that they've had TB in the past, but who have chest x-ray scars of that. And actually, I originally put this analysis together because that's kind of a funny category, and we'd like to understand more about that. And the first question was, is that even a real category? You know, there's other things that can cause scars on a chest x-ray. Do we really believe that that is the radiologist, who's of course blinded to everything, but is he making this up? What does he see? So we put this together, and, you know, I'll be interested to hear what you all think. But for me, looking at these HIV negative people, I mean, this was really quite interesting. It does look convincing that, you know, with age, there's more and more lifetime experience of some kind of TB, but the kind of distribution of what kind of TB is kind of striking, in that most people are this orange color. And actually, it's really interesting that even in such a high transmission area, really that old adage of about 10% of people having active disease in their lifetime seems to be true in the HIV negatives. But then if we look over at the HIV positives, we can see that, first of all, at every age, there's already more TB. This highest amount in these young people is probably because of perinatal transmission. So these people have probably been living with HIV longer, actually, than the people in their 20s. But also we can see there's kind of a strikingly different distribution of TB phenotypes among people who do have TB experience, with many more people with HIV having had disease requiring treatment. And, you know, if we think that we're looking at here, perhaps people who are immunologically resolved their TB themselves and are maintaining a state of non-infection, versus people who HIV tipped into loss of control of their infection into disease, you know, I think that that's what this looks like to me, and quite eager to start to look at biological correlates of that in the samples and to try to study that more. So very briefly, I just want to show you one more aspects of the kind of way that we can look at this data. So this, I'm just going to show you a couple cases that will give you a sense of the kind of detailed phenotype that we have here. So this person is a 59-year-old woman who's HIV positive. She's been on antiretroviral therapy for quite a long time. She's had TB 3 times. And then she actually comes to Vukuzazi on TB treatment. So this is her 4th time. And, and she, her sputum reflects that she's on treatment. She's gene expert positive, but culture negative. And we can see that she really has quite a socked in both scarred, old scar, and active consolidation and cavitation of this right upper lobe. And she has disease in the left lung as well. But she was on treatment. And now she fails treatment again. You know, she could need surgical evaluation. But at the moment she was on treatment, and so she needed just to continue that. Then there was this younger woman, HIV negative, who has a left upper lobe consolidation, but she's had TB once in the past. Her sputum was negative by both gene expert and culture. And so the medical officer brought this case to my attention, and we agonized over this one a little bit. But we decided at the time, well, we couldn't rule out that this was due to old disease. And so, because she was asymptomatic, we thought she didn't need treatment right now. Although, of course, one would want to monitor her. And then there's this 17-year-old young man who's HIV negative and has this left upper lobe consolidation. And he was also asymptomatic, but had positive sputum, both gene expert and culture, so consistent with active TB disease. And all of these cases are interesting, and we see them as individuals. We've looked at the data aggregate, as I've shown you, but what it starts to get really interesting is we realize that, you know, and we haven't fully gotten into this, but these 3 cases all live in the same household. And then there's actually 3 people in this household who do not have evidence of tuberculosis. And very briefly, you know, interesting features that this demonstrates, first of all, like most people in the cohort, people are living in sero-mixed households. In this particular household, there's been quite a lot of TB, and one wonders if there's something genetic there, or perhaps they just have never managed to clear it from the house. It's just being passed around. But the other striking thing about this household, this household, is that everyone was asymptomatic. So if you think about kind of screening, that's just quite striking. And that's been 80% of people with microbiologically proven active TB that we've picked up in Vukuzazi are absolutely asymptomatic. And we wanted to understand how these people actually related to each other, and it actually, in a demographic surveillance, we didn't, the demographer was able to say, I can tell you how they feel they're related to each other, because that's kind of demography methods. But I said, well, I'd really like to know how they're actually related. He was able to figure it out by using birth records, etc. And we could see that this woman is the grandmother, that she lives with, you know, she's had 6 adult children, 4 of whom are alive, mixed HIV status. We can see their chest x-rays here, and then this is a grandson. And then, of course, there are other children in this house. They aren't eligible for Vukuzazi, but of intense interest for both transmission and their own health. And we have a R01 that's close, we hope, to be funded to actually start to characterize and treat the children in these households as well. So last slide. Where are we going with this? So of course, we're currently trying to understand the determinants, transmission patterns of TB in the population, looking at these overlaps as we described, as we set out. But very quickly, we want to move towards trying to understand better this immunological heterogeneity that we see between people, trying to understand that both in the peripheral blood and in lung, and then to rapidly move towards testing strategies to interrupt TB transmission by interrupting progression of MTB infection to TB disease. And the idea is to be able to actually move up and down these levels of study. This is a large study team, obviously, and particularly I'd like to mention Alison Grant, who many of you may know, who she and I really focus on TB in this population and in Vukuzazi. Kathy Baisley, who's the statistician who made some of the slides I showed you, the population epidemiology slides. And then this is, just at the beginning, I showed you a little bit of data from my lab in Durban, which this is just a couple of the members of. So I will stop and hand over to
Great.
Greg, yeah.
Good. So wonderful, fantastic presentation, really impressive. Just a reminder to others, just text me. You can see a chat window. Just text me if you have a question. All you have to do is say I have a question. You don't have to give me the question. I'll just call on you. I already have a couple. Emily, I just wanted to start it off. You know, it's fantastic, incredible work enrolling all those people and getting, I think, a pretty good enrollment rate, given all the data that you're collecting. I just wondered, so is this area near any mines? I also had a question about, are you doing any sort of evaluation for latent TB among all those cases among the 10,000? I mean, over time, you're going to have some TB cases that are incident if you did do that. And then finally, just one comment, you may be seeing those HIV negative patients with a history of TB diagnosed by chest x-ray because they have, being HIV negative, perhaps have more scarring and more lung damage than the HIV infected individuals. But I'll start with those 2 questions, and then we have a question from Neil Gandhi.
Okay, great. So the first question, very good. the question about the mines. So first of all, we know that a lot of the men from this area actually have historically migrated to Gauteng for work in the underground mines. And we haven't started to analyze the TB data that I've shown you in light of that, but it's on the list. But interestingly, just in the last couple years, there's a new open-faced coal mine within the demographic surveillance area. And we do already know that that's an HIV hotspot, unfortunately, HIV transmission hotspot. And we, of course, are quite interested in its impact on respiratory health, both for people actually working there, but also just the surrounding people. That's one of the environmental exposures I mentioned. The second question about latent TB, you know, we thought long and hard about this, and we didn't think it was practical to do latent TB testing on everyone. We had hoped to do it on a kind of random selection of people so that we would get some kind of a baseline rate, especially as a baseline measure if we start to do interventions that we aim to decrease transmission. And we did do separately a small adolescent incidence of latent TB infection cohort that could serve that purpose. But part of the idea of doing this very quickly is that now we can hopefully identify subgroups of people over specific phenotypes who are of particular interest, and then we can go back and do additional further characterization of some of those people, and latent TB, collection of PBMCs, et cetera, even bronchoscopy are on the wish list for those kind of things. And finally, yes, I think your point, absolutely, I think we're seeing more lung scarring in the HIV positives, and the general question of kind of the impact of the lung scarring on long-term health, I think is really of interest, and actually not something I'd love to have us working on, but no one's actively working on it right now.
Okay, great, thank you. Neil?
Emily, this is Neil Gandhi from Emory University. Congratulations on this fabulous cohort. It's really exciting to see, and I can only imagine all of the wonderful work that's going to come out of it. I actually have two questions, and they build a little bit on Greg's questions. So the first was how migration, like to the mines, influences whether somebody is included in the sample or not, or how does somebody need to live in the area full-time to be recruited into the sample? And then my second question was related to your HIV negatives who don't self-report TB, but you're diagnosing possible previous TB on chest X-ray, whether you think that that might represent an episode of subclinical TB they may have had, and could that have been a time when they were infectious and part of the transmission cascade?
Yep. No, thank you for those excellent questions. The first one, so we do say that the person has to be resident in the area to be eligible for Vukuzazi at this time. And that means, I think, they have to sleep there for like at least a month in a year, but also practically they have to be around for us to, enroll them. In our fantasies, we talk about, at the end of this time, it would be really cool to send a Vukuzazi van to Durban, Joburg, and actually try to enroll our non-resident members of the population. But that's on the wish list. But we have kind of talked about trying to do that, but that's the answer to that question. The second question, I think that's absolutely what I'm thinking is probably happening. And as I said, we didn't have time to really go into the people who have active TB because we just are starting to get all those cultures matured to look at now. But we certainly are seeing high rates of asymptomatic TB, which, by some definitions, you could say many of the people that we're picking up in the population are having subclinical TB, depending on how you define subclinical. And some of them could be destined to re-control if we hadn't come around and snapped a chest X-ray and taken a sputum. Maybe they would have naturally controlled. But certainly many of them, or all of the ones I'm speaking about right now, these microbiologically proven ones, are able to cough up organism. So we consider that infectious TB. So I think that's part of what it seems that actually the people with symptoms, and especially all these people with HIV who go to clinic all the time, we're starting to see, because Allison was actually doing a clinic prevalence survey at the same time that we were doing this population prevalence survey. And we actually found similar rates of prevalence of active infectious TB. So it almost seems that the clinic going population is pretty well screened for TB. The remaining missing cases are out in the population. A lot of people HIV negative, most people asymptomatic. And yes, I think that is contributing to ongoing transmission. So those are some of the ideas we're thinking about in terms of how to now intervene and stop transmission.
Great.
Great, thank you.
Karen Jacobson.
Hi, Emily. It's Karen Jacobson from Boston University. Really exciting study. I think I'm working in the Western Cape, and on a much smaller scale, I'm seeing similar patterns that is striking and a lot to explore. Two questions, one building on what Neil just mentioned. It'd be interesting to know who's getting, if you're looking in the clinic and then you're looking in the community to capture who's being missed. I think historically it's often been seen that young men are those who least access clinics, and so any of our interventions that are looking for TB mostly in the clinics will miss them. But it sounds like you have an opportunity to really try to quantify again if there's additional targeting beyond the clinics, who are these missed populations for TB screening. And then second is, it looks like you have a real opportunity again to do some exciting molecular epi to understand a transmission when you see co-prevalent cases in the household when you're in these endemic settings. I think what I struggle with is how often we see discordance within the household and then saying that means that they aren't related to each other. I think that we need to get more advanced in how we understand strain relatedness, especially in places where people probably have had multiple infections over their lifetime. But at least from what you describe, if you have all of these households and you have a sense of relatedness, I imagine that that's something you guys have set up for yourself. So I'd be interested to hear more. So now I'll go quiet. Thank you.
Thanks. So your first question, yes, I mean, this is what we're, trying to figure out where are the remaining TB cases, because I don't know if you guys saw just actually today, someone from, it came out in Scientific Reports led by Frank Tanzer from our historical data from from reported cases in the clinics from our demographic area. Case reports in the clinics, those rates are going down with the increased spread of antiretroviral therapy. But so now we're really on the hunt for the missing cases, as you say. I do think it's men, it's HIV negative people, it's asymptomatic people. In terms of coming up with targets and people to screen, I think we have some preliminary ideas, and we as I say, we'll get into that data in a little more detail as it gets complete. And that will be also interesting. This first 10,000 are really in the rural part of our demographic surveillance. And currently we're enrolling in the urban, the kind of peri-urban part, and the rates are even higher there of TB. But there may also be differences between those two settings. We are finding a lot more kind of working people in the peri-urban area and people starting to gather in different places. And that'll also be, as you say, very interesting from the molecular epi side. We've just now got about 150 strains of active cases, so we're going to start sequencing. But I anticipate, as you say, and as Neil has shown, it's going to be very confusing. And we look forward maybe to, talking about those issues, because, if we see similar to what you're describing, it's this seemingly related people having completely different strains at the same time. I also don't know how to really wrap my head around that, but we haven't actually started sequencing yet.
Good. We have a couple more questions. Dana Black.
Hi, this is Danae. Thanks, Emily, so much for your presentation. I had a quick question about TB preventative therapy and if it's being used in the area that you're working in, and kind of what the utilization in your population is like if you're collecting that information.
So great question. It is technically recommended in our Department of Health guidelines for people with HIV for whom active TB has been ruled out, and of course for children under 5. Practically, it's inconsistently given at the clinics among HIV positive people, but we do have a pretty good system linked to pharmacy databases, et cetera, so we can analyze that. There has been an ongoing small, like community-based trial of community-based IPT and ARV, the DUART study, which Adrian, who's on the call, I think just presented the first results from, that's in parts of the demographic surveillance area. So we'll have to take that into consideration when we get to those geographic parts of the surveillance area, and they should be able to give us, we should be able to link up that information. But I think, you know, in terms of thinking about interventions, again, some kind of preventative therapy, perhaps one of the newer short course would be also on the list of things we want to probably do as an intervention going when we get to that point, which will hopefully be soon.
Okay, excellent. I think we have the last one here, Adrian Hero.
Thanks so much, Emily. So exciting to see this data from Vukuzazi. It's really, just let me echo that what everyone else had, that it's tremendous work getting everyone enrolled and questioned and surveyed. And I know what a huge lift it's been. So congrats. I realize that things are still in maybe somewhat of an early stage of analysis, but I'm curious to know if you have any sense of TB, to what degree TB is clustered in households, meaning especially knowing that there's pretty good clinic detection and prevalence reporting among people who do show up at clinic. Are most TB cases found located in households where there is a known TB case already, even if the case that's found from the community screening has not been previously known? Are they more likely to be in a house where there is one person, such as the household example that you showed?
Yeah, so as you say, I don't have a formal analysis of that yet, but no, that's not very common. We have, you know,
Lots of solo households.
Yeah, we have some interesting households like the one that I showed you, but most people are not living with a known active case.
Thanks so much. You know, obviously implications for how effective household contact tracing is going to be for transmission interruptions. So fascinating to know. Thank you.
I think that's what came up on the last call, and I said that, you know, that was the topic of the last one, and that's where this relates. Yeah.
Wonderful, Emily and everybody. Thanks so much for the active participation, and Emily for presenting. It's really a fantastic study, and can't wait to see over the next several years what comes out of it. We had almost 50 people on the call today, I think, and so you know, many people who are leaders in HIV/TB research all coming together. It's very, very exciting. So we can stop now because it's top of the hour, and you'll be hearing from us about the next call, which we'll schedule for the near future. Thanks, everybody. Have a great weekend.
Thanks, Greg.